Hello, everyone. I want to begin by telling you a story of a silly man. This silly man was asked by one of his followers to make a video about barnacles. Now, this man knew virtually nothing about these enigmatic creatures, and so dedicated an entire evening, one whole evening, to Googling barnacle and barnacle reproduction. Upon this epic journey, he relied upon only the most reputable of sources, such as encyclopedia.com and Wikipedia. And at the end of this exhaustive evening, he came to the unassailable conclusion that all barnacle researchers since Darwin were wrong. Barnacles were obviously independently created. They couldn't have evolved. That all barnacle phylogenies are just a farce, a fairy tale, a made-up story that you tell children. And like a Dickens miracle, he had uncovered this fact in but a single evening. So convinced was he of this truth that he issued a challenge to all evolutionists to respond and demonstrate to him the evolution of the barnacle. It goes without stating that I don't take Kent Hovind very seriously, because I frankly don't think that he takes science very seriously. I think that he sees evolution as just a punching bag, as a way to keep people engaged and watching his content and to keep himself relevant in a world that has largely left him behind. But apart from all of that, I have decided to actually answer Kent Hovind's challenge and explain to you today the evolution of the barnacles. So sit back, relax, and prepare for an epic dive into one of the strangest crustaceans you're likely to see. So this is going to be the layout of this presentation. I'm going to start off first by giving you a primer on evolution and evolutionary models. Next, I'm going to introduce you to the diversity of the barnacles, i.e. the cirripedia, and their relatives. And then I'm going to talk a little bit about how barnacles are different from other crustaceans. So obviously, to demonstrate that barnacles are actually just evolved crustaceans, we need to explain how those specific traits evolved. Then I'm going to give you a proposed evolutionary model of barnacles, and then we're going to see if we can find some support from this model from things like comparative anatomy, developmental biology, and finally, molecular biology. And then I'm going to end with just a little bit of musings about the philosophy of science, because more often than not, it seems like creationists are very confused about what science tells us, how it tells us things, and how to interpret results in a scientific framework. So I think it could be useful at the end here to put a little bow on this by just talking about the philosophy of science. First, a primer on evolution. This is very important because more often than not, it seems like creationists don't have a very firm understanding on what evolution even is. So it's very difficult to go down a path proposing an evolutionary model when you don't really even get the base theory, right? So let's first give a little primer on what evolution is. So succinctly, evolution is inheritance plus change in populations. A rather famous biologist put this as descent with modification. Now, we understand the mechanisms of inheritance as being described by Mendel's laws and the material of inheritance as being DNA. Now, evolution contends, evolutionary theory holds that those things, Mendel's laws of inheritance and DNA as the material of inheritance, aren't arbitrary, that those things have always been true, that Mendel's laws have always worked, DNA has always been the material of inheritance. That's an important contention of evolutionary theory, and observations have thus far confirmed this. We've never seen an instance in which Mendel's laws don't work or DNA is not the material of inheritance. Now, secondly, is the change component, the modification aspect. This is that heritable characteristics in populations are not static. they change through time, right? Populations are not these constant things. And we can theoretically propose mechanisms that cause this change. These include Darwin's mechanism of natural selection, genetic drift, gene flow, and mutation. These four mechanisms can alter the heritable characteristics of populations. And we've evaluated this in both an analytical framework, that is a mathematical analytical framework. We've evaluated them statistically, and we've applied those statistical models to actual natural populations. And we have consistently verified that these mechanisms will produce predictable changes, right? Now, evolution also supposes that this change has always been true, that it will be true in the future, populations will change, and into the past, populations have changed. That this is not arbitrary. At no point in the past did populations suddenly not change, right? So again, observations have thus far confirmed this. We have never observed a population that just doesn't change, right? That is incapable of changing. Therefore, for evolution to be true, then inheritance, descent, and modification or change must have been true for all points in time into the past. These two things aren't arbitrary, right? That's the key to understanding universal common ancestry and why we accept it as a fact. So, for example, if I give you a phylogenetic tree of closely related species, right, the mechanisms of inheritance explain that, just as it does with subsequently deeper and deeper and bigger trees, right? Such that at no point in time did the mechanisms of evolution or the mechanisms of inheritance just stop working. And we have to draw a line on a phylogeny and say, okay, everything back here is not true. Evolution ceases to work. Inheritance ceases to work beyond this point. But this way, DNA is the material of inheritance and does reflect ancestry, right? That is an arbitrary line that has never been observed. We do not know any mechanism that would generate that arbitrariness to descent with modification. To claim that inheritance or change are arbitrary is to invoke an unobserved, untested, completely new theory. Therefore, the burden of proof is on you to demonstrate where this point is, where this break between our current understanding of inheritance and descent with modification is, and your new proposed mechanism that explains the back half of phylogenetic trees, right? So that is why we fundamentally accept universal common ancestry as fact. Now, let's talk about evolutionary models. The reason I give that background is because, importantly, the fact of universal common ancestry, that is to say that all barnacles share a common ancestor with all the rest of the crustaceans, that share a common ancestor with all the rest of the arthropods, etc., does not rely on the accuracy of any proposed evolutionary model. We can have absolutely no idea how a new trait evolved, and universal common ancestry is still a fact. It's still a fact because we understand how inheritance works and we understand the mechanisms that cause change in populations. That's all you need for universal common ancestry to be fact. So evolutionary models, what they try to do is they are hypotheses that try to explain the mode and tempo of specific kinds of changes within a very specific taxa. Importantly, like all scientific hypotheses, they are tentative, they are testable, they are falsifiable, and they are predictive, right? They are used to predict some future observation that we haven't yet seen. They can be falsified by one of those future observations, and there's something we can test. We can actually test this out by either sequencing individuals, digging into the dirt, looking into their development. There's ways in which we can design experiments to actually test this hypothesis. Importantly, they are not true. They do not represent any kind of absolute truth. okay? They are just models. They are models to try to help us understand how a lineage could have arisen from ancestral traits. We propose that model, and then the accuracy, inaccuracy, it all boils down to how much support does that model have from the material evidence, right? They are not true in any kind of metaphysical sense. We will likely never know the exact way barnacles evolved, but we can put forward very good models to try to predict and explain how it could have happened. Testable explanations is a fundamental part of science. Now, with all of that background out of the way, let's dive in and actually talk about barnacles. So barnacles are fascinating little crustaceans. They have long been one of my favorite critters because they are bizarre. They have a myriad of different shapes and sizes and life history strategies, and they are just really, really fascinating critters. They also fascinated Darwin, famously, who spent 8 years studying these little guys. And in fact, his 1854 monograph on the cirripeds is still the go-to monograph for the systematics and taxonomy of barnacles. So if you are a barnacle systematist, you've probably read his 1854 monograph. It's still that important. So let's talk about first the diversity of the Thecostracca. Thecostracca means case shell, and it's a class of pancrustaceans that includes all of the things we call barnacles, as well as things that aren't quite what we would refer to when we say barnacles. It includes things like the Facetotecta, the Ascotheracida, and the Cirripedia, that includes barnacles, as well as things that you wouldn't immediately recognize as barnacles. Now, I want to start with this kind of broad scale diversity because Kent, in his night of epic Googling, came to the conclusion that there were only 2 kinds of barnacles, acorn and goose. There's a lot more kinds of barnacles than that, and that's a pretty poor indictment on your Googling skills if you only came up with those 2. This is the phylogeny that represents family level diversity. So the tips of these are families, which means there's genera and there are species that are far more diverse even within those groups, okay? The goose barnacles represent just one small part of the Thoracicans, and the acorn barnacles are just one infraclass within the Thoracicans. It also completely ignores Rhizocephala, which are very much barnacles. They don't look like barnacles. They're really, really bizarre, and we'll talk about them, but they are a part of the cirripeds and can't just be ignored. So let's start off by talking about the anatomy of barnacles. Barnacles they look really strange. They seem completely alien. How can we even begin to understand their evolution when their just base anatomy is so bizarre, right? Well, the way I want to approach this is actually first talking about the anatomy of a shrimp. Why shrimp, you might ask? To paraphrase Louis Agassiz, barnacles are just shrimp that stand on their head and kick their food to their mouth. So let's look a little bit into the anatomy of a shrimp, okay? Now remember, barnacles are crustaceans, and so they should have many anatomical similarities to other crustaceans, including shrimp. So let's break this up. The front here is the cephalothorax. This includes the head region and the thorax region. In a lot of crustaceans, the head and the thorax have been fused, but not in all. Some of them, these are actually 2 separate structures, but in a lot of the, especially Malacostracans, they've been fused together. The back section is the abdomen. Now, there's 3 important anatomical features of shrimp that I want us to really focus on so that we can begin to orient ourselves with the anatomy of a barnacle. So, starting first, note where the mouth is here, that it has a very short esophagus that leads to a large stomach, okay? So remember the structure, and remember that the stomach is dorsal. That's to say that it runs along the back instead of in the front. So humans, our digestive system is ventral instead of dorsal, and our nervous system is dorsal, right? But in crustaceans, it's flipped. So the stomach and the digestive tract are dorsal to the nerve cord here, which runs ventral. All right. So in barnacles, what we expect to see is that they should have a ventral nerve cord and a dorsal digestive tract. Furthermore, notice importantly the position of the gonads. So this is a picture of a female, but males, this would be the testes and the ovaries would be in the same place. The ovaries sit on top of the digestive tract. And the testes also would be sitting on top of the digestive tract. And then notice that the gonopore has to cross the digestive tract and the nervous system to then exit ventrally, right? So these features are very important. First, the nervous system is the most ventral, then the digestive system, then finally the reproductive system. So we should see this three-layered system in barnacles as well. Another important feature to note about barnacles is that they don't have an abdomen. So I've drawn a little line here showing where that would be cut off. So they have the thorax region and the head, but they completely lack an abdomen, which is totally bizarre, and we're going to talk in detail about how they lost that abdomen. Okay, now let's see how the barnacle's internal anatomy compares to that of the shrimp. So in the center here is the barnacle. Sorry about the granulated figure. It was just the best one that I could find. And then up here on the left is that same shrimp. I've just flipped him upside down and removed the abdomen. So let's see if we can orient ourselves with the internal anatomy of the barnacles. So starting here, this is the entrance to the operculum. We'll talk about the shell anatomy in a moment, but let's just focus internally. Entering here, these are the thoracopods. In barnacles, they're called cirri or cirripeds, but they're homologous to the thoracopods of shrimp. So if you look over here, these are the legs that we're looking at. That's what these legs are here. Next, we get to the mouth. So here's the mouth from the shrimp. Here is the mouth of the barnacle. Very short esophagus, just like in the shrimp. And then we enter into a large stomach. There's the large stomach in the shrimp. Notice that the nerve cord should run right across the stomach. This little blue structure right here, this is the nerve cord as well as the ganglia. You can see it innervating each one of the different legs. Let's follow on back. We get down into the digestive tract. Now remember, the barnacle is turned upside down, right? So the digestive tract in shrimp are all dorsal, with the nerve cord being ventral, and it's exactly the same in barnacles. We've just turned the orientation up, right? So the ventral side is facing up instead of down as it does in shrimp. So again, here's the digestive tract. Now sitting just dorsal to the digestive tract is the gonads. So here are the ovaries in the shrimp. Note here are the testes in the barnacle. Now, barnacles, importantly, the Bilanomorphs and many of the gooseneck barnacles are hermaphroditic. So that means that they have both sex organs present in each individual. And so what they've done here is we have testes that are sitting just posterior to the ventral, to the dorsal digestive tract, and then the ovaries sit just behind that here. Now note that they both have to exit ventrally, which is fine for the testes. It just follows the digestive tract up and exits. But notice what the female gonopore has to do. It wraps all the way around, goes out across the stomach, and then exits ventrally. So they both have to exit ventrally. Since the ovaries are behind even the testes, the gonopore becomes exceptionally long to compensate for it. But again, just like in the shrimp, we see that the nerve cord is ventral. It runs along the legs, along the thoracopods. The digestive tract sits just behind it, and then the gonads sit just behind that. So again, as we can see, barnacles are just upside down shrimp that have been planted in by their heads. Now let's talk about the shells. So the barnacles are really important for having both inner plates and outer plates. This is true in the Bilanomorphs, not true in the Scalpiforms, which we'll differentiate those in just a moment. So first, let me orient you a little bit. When we're talking about the outer plates, barnacles sit in a outer plate, and then they have within it the actual living critter that is also surrounded by plates. So there's like 2 layers of plates. The outer plate is immovable. Okay, it's cemented, it doesn't move, and it's made up of a rostrum, which sits in front, a carina, which sits behind, and then laterals that sit on either side. Now, these plates are fused, and so it's difficult to differentiate them. For this figure, I just drew like some white lines here to kind of help you see where the fusion plates are. So this is a top view, and then this is just flipping it over and looking at it from the bottom side. And then on the bottom, I've actually included the inner movable plates. So these are paired tergum, which sits behind, and scutum, which sits in front. The tergum actually come up in little triangles, and they kind of sit behind it like this, and then the scutum sit within the tergum. Okay, and those plates can kind of shift and move around, and then they open up. The operculum opens and allows the little cirri to come out. So that's kind of the basic shell morphology of at least the Bilanomorphs. Again, the Scalpomorphs are a little bit different, and we'll talk about them in a moment. Okay, so how do you get a barnacle? What is the basic barnacle development? So barnacles develop really, really curiously. All barnacles start off as a nauplius, and that's this little critter here. Now, nauplius are pretty fascinating in that many crustacean groups have a naupliar larval form. Okay, they are free swimming, float around in the plankton. Some of predators, some actually have little yolk sacs. They're lithotrophic, and so can feed themselves for a while they're floating around. But interestingly, you can't tell for most crustaceans what the adult form is going to look like just by looking at the nauplius, right? So copepods have these, various other crustaceans have these. Barnacles you can tell apart solely because they have what are called the frontal horns. So these little protrusions here, and then this is a scanning electron micrograph. You can see them here. Barnacles are the only ones that have these frontal horns. We don't know what the function of them is. They appear to secrete things, but we don't know what the secretion is. So we don't actually know what the function of the horns are. But barnacles are the only ones that have it. So that is a unique character. So whenever you see a nauplius, like under a microscope or from a sample, and you see these little horns, you know the adult is going to be a barnacle. You don't know what kind of barnacle. It could be any one of those groups that I just talked about, but you know it's going to be a barnacle, right? So the nauplius goes through 6 different molting cycles. It starts off looking just like it is, and then every molt, it grows a little bit bigger until eventually it metamorphoses into a cyprid. And I'm going to talk about where the name cyprid comes from, but I don't want to spoil it yet, so I'm not going to tell you. But it's basically a bivalved crustacean that has a thoracopod, which is this structure that you see here. This is its thorax, that it just extends it and does this kind of kicking motion. That's how it swims around. And then it has these 2 very robust front antennae. Okay, so there those are. And then what the cyprid does is it finds a settling area, and it detects the area that it wants to settle by hormonal cues that are given off by adult barnacles. So basically, it swims around, it comes upon an area that it wants to settle, it will land on it, and then it may or may not settle there. In fact, a lot of cyprids won't settle at the very first place they land. But if they land and they begin to pick up adult hormonal cues from around them, then they'll decide to actually settle. And the idea behind this is that you don't want to settle in a place in which there are absolutely no conspecifics for you to mate with. And it might also indicate that the place is not a very safe place to actually settle because there are no more barnacles around, right? So you want to be able to discriminate. So they do this again by detecting adult hormonal cues. And then they land, they shed the carapace, and then they begin to form into the adult. And we're going to look at that in a little bit more detail. So this is a really cool paper that had this time-lapse photographs of this settling process. So first they start off as this cemented cyprid. So the cyprid lands, it cements its antenna onto the substrate. You can see that it still has a compound eye at this point. I should have mentioned before that cypripeds have a compound eye, which is a fusion of the naupliar eye. Next, they begin metamorphosis. And what this metamorphosis does is it actually separates the tissue that's holding the carapace on, right? It separates that tissue, and it separates the tissue that's holding on its stalked compound eye. So phase 3, what it will literally do is it will lift itself up. So it's getting itself into the orientation that the barnacle normally lives in. And then it starts to do this shaking motion, right? It just starts shaking. And what it's trying to do is it's trying to slough off that carapace. And it literally will just shake that carapace loose until it flies off. And not only the carapace, but also its compound eyes. It literally shakes its eyes and carapace off. So here's that feature. You can see this is the carapace cuticle just kind of being sloughed off. Here's the compound eye being sloughed off. And then all that's left is this little globular critter that has now shed its carapace. Now, it does still have an exoskeleton, importantly. It's not like it's just completely naked, but the exoskeleton is very soft, right? It's not strong, and it's not well protected like it would be in a carapace or in the adult form when it has the plates. And then finally, once it's completely settled, it begins to form the basis, which is like the plate that's either in the Bilanomorphs, going to stay relatively small, or in the Scalpomorphs, it's going to actually start growing into the peduncle, right? And because remember, they're stalked. I'll show you some images of them in a moment. And then it slowly begins to add plates in a sequential manner, right? So that's the basic development of a barnacle. And then this is some scanning electron micrographs of the early stages before it has started laying any of its primary plates. You can see the basis is still here. Again, like I said, that's going to grow into the peduncle and the stalked forms, but it's going to stay pretty much that size in the Bilanomorphs, the ones that are sessile that don't have the stalk. Interestingly, these are called spats. I don't know where that name comes from, but they are commonly a juvenile barnacle that has settled and is no longer a cyprid is called a spat. Okay, so like I said, this same Cyprus form can lead to any barnacle. Every one of the major groups of barnacles come from this same developmental pathway, which is a pretty fascinating feat and is a very, very strong indication that all barnacles have a common ancestor. They follow this exact same developmental pathway. So now let's actually talk about the taxonomic diversity of specifically the Thoracicans. We're going to include the Rhizocephalans as well, so I should broaden that and say Cirripedia. But the first two we're going to look at are the Thoracicans. And these are what Kent called goosenecks, or goose, right? So all of these contain a large peduncle. The more ancient family, you can call they're often called more primitive. What that basically means is that the modern descendants retain more of the ancestral characters than some comparative relative does, right? So to compare, like, for example, the Scopelioids with the lapidoids have a lot more derived characteristics. The Scopeloids look a lot more like what the fossil forms look like. So that's what we mean when we say primitive. Lepus anatifera is one of the lapidoids. It's a very common gooseneck barnacle. If you've ever been to a beach, you'll find these little guys attached to just anything, bottles, seaweed, etc., etc. They all only have 5 plate valves, which this is considered a derived characteristic. A lot of the scalpoids actually have tons of little plates. You can see them here. There's lots of them. They are just kind of scattered all around. Some of them are seemingly random. There's not a lot of order to where you see them, right? There's not a lot of order, and they can take on all kinds of weird and funny shapes. Whereas the lepids, they all are like fused together, very tight, and form a very neat shell. An example of that is this one here, where you can see they fuse the sides and you get a very nice watertight enclosure. Whereas the scalpoids, again, they're the most diverse of all the cirripeds, and they can have up to 14 shell plates. Curiously, they are also have a very interesting sexual system that includes androdioecious as well as being truly dioecious. So androdioecious means that you have a normal-sized female and then typically a dwarf or parasitic male, whereas truly dioecious is what we know as just completely separate sexes, where the males and the females look roughly the same. The Bilanomorphs are the more recognizable barnacles. These are the things you see on rock jetties in the intertidal zone. They're one of the most abundant and considered one of the most derived. They're the most different than what we think that the ancestral barnacle was like. Importantly, they don't have a peduncle. They still have the basis that becomes the peduncle in the Scalpelomorphs, but here it just doesn't grow. They stay flat. They are enclosed within articulating plates that sit within a plate wall. So this goes back to the rostrum, carina, and lateral that I was talking about in the previous, as well as the tergum and the scutum that sits in front. That's what we're referring to by outer plate and inner plate. The cathameloids are incredibly abundant, especially in intertidal zones. If you've gone down to a rock in an intertidal zone, you've probably seen just gazillions of these tiny, tiny little bitty barnacles. The common name of those are buckshark barnacles, and they're members of the cathameloids. So they have either 8, 6, or 4 plate walls, and yeah, they're considered again a little bit more primitive because they have a little bit more variation in the plate wall number, whereas all the other Bilanomorphs only have 4 plates. Balanoids are the bigger barnacles, so like Balanus, Megabalanus, the acorn barnacles. These are all typified by balanoids. Again, they only have 4 plate walls, and you can find these not at the higher intertidal zone. They typically occur a little bit further out in the water, so they need to be submerged more often than the buckshark barnacles do. Lastly, we're going to talk about the Rhizocephala. The rhizocephalans are highly derived parasites that largely infect decapods. Rhizocephala are barnacles, but they don't look anything like barnacles. The adult form are completely unrecognizable to other barnacles. So you can see these little pouches, these little extensions that you see on the decapods here. Those are all rhizocephalan parasites. Let's talk about how exactly this works. They are really, really bizarre. So a female cyprid, which the cyprid looks just like it does in all other barnacles, it lands onto a host and it burrows into its gills. And then it grows this root-like structure inside of it, inside of its host. And this is called the interna. And I'm going to show you what this root-like structure looks like. But it basically, if you were to see it, you might think it was like a fungus. It's kind of filamentous, and it just grows into all the parts of the crab. And it does this because it's just going to absorb the nutrients that exist within the body wall. Just absorbs it. Eventually what happens is that as it reaches sexual maturity, an externa will burst forth from the host. That's the thing that you're seeing here, here, and here. These little structures, that's the externa. And the externa is going to be fertilized by a male cyprid. Now, rhizocephalans are really, really bizarre in that they are what's called parasitic castrators. So whenever a rhizocephalan parasite gets into a male crab, it will castrate it, and then it will take over its physiology, and it will make it behave and take on morphological characteristics of a female crab. Now, in male crabs, they have what is called, in the abdomen of a male crab, folds under it like this. So if you've ever seen, right, a shrimp, the abdomen sticks out, but in crabs, it's just folded and fused ventrally. Well, the males have a very, very thin abdomen because they don't brood offspring, so their abdomen is very thin. The females is much wider because that's where they keep eggs. They brood them within this folded under abdomen, and when they're ready to release the eggs, they just kind of flap it like this. Males don't have that. But when they're infected with rhizocephalan parasites, their little narrow abdomen will expand, and the externa will just expel from right there where it would be a brood chamber in a female, and the males will then take care of the eggs as if they are female. They will even flap the abdomen like this when it's time for the externa to be fertilized, and this will attract male cypripeds to come and fertilize the parasite. Really, really bizarre barnacles. So this is a stained image, a stained 3D image of the externa, which is colored this like it's the pea looking thing. Looks like a little bean, little pea here. And it's colored orange, and then the interna, the root system, is colored green. So this is a hermit crab and an anamurin, and you can see the externa expanding here from the abdomen, and then inside of it is the root system that has just infested all of the abdomen and has even started to crawl up into the thorax. So Rhizocephala, absolutely bizarre, but really, really cool barnacles, and I'm disappointed that Kent did not bring them up. Okay, so how are barnacles different from other pancrustaceans? This is one of the things I talked about before, that if we're going to talk about the evolution of barnacles, we need to explain where these derived characteristics come from, right? Characteristics that are quite a bit different from the crustaceans that they're related to, right? So what are the big things we need a model to try to explain? One is the sessile lifestyle. There are some sessile crustaceans, but barnacles are the sessile crustacean, right? So we need to explain how that arose. Secondly, the lack of an abdomen. This is really weird. Why do barnacles not have them? What happened to that abdomen? Third is hermaphroditism. So very, very few crustaceans are true hermaphrodites. They may change their sexes throughout their lifespan, but they're generally only one sex at a time. Whereas barnacles, especially the Bilanomorphs and some of the Scalpelomorphs, are true hermaphrodites. So we need to think about how that arose. And then finally, the formation of the shell plates, right? That enclosing structure that is so characteristic of barnacles, but that does not exist in other crustaceans. We need to think about how we can explain that evolutionarily. Now, with all of that background information in place, let's propose an evolutionary model of barnacles. Now, I don't want to lead you to think that this is somehow my model. Really, this is a Frankenstein of a bunch of models that I've read about and just kind of conglomerated into one so that I could present to you and we could kind of talk through how this works. So here's my model in cartoon form. Let's talk through it. So first off, our hypothetical ancestor of barnacles was probably a free-swimming dioecious crustacean that had a carapace, that had a thorax, that had an abdomen and paired antennae. So that's our hypothetical ancestor. The reason that the intuition for this comes from the fact that barnacles are most closely related to copepods and malacostroking crustaceans, both of which are largely free-living, free-swimming, and dioecious, and that have thoracopods and an abdomen. Barnacles appear to be very derived from that, so it's more likely, i.e., it's more parsimonious, that the ancestor was free-swimming. Next, what that ancestor must have done is planted itself onto a substrate. It became sedentary, right? But it would have retained most of those ancestral characteristics, including things like the carapace, the thoracopods, and the abdomen, and probably was still dioecious, right? However, lived most of its life sedentary, and then maybe just did some feeding with its legs. From there, we then need to move into a step of getting rid of that carapace, right? Because all adult barnacles lose their carapace. So some ancestral barnacle must have lost that carapace. How did they do that? How did they lose their carapace and manage to not just get eaten while being exposed, stuck to substrate? Well, a good possible way in which they could have got around this is to actually burrow into the substrate. Right? So if you can burrow into the substrate, you can shed that carapace, be protected by the substrate, and still continue feeding out of it. So you didn't need to start building shells immediately if you can just get protection from the substrate that you live within. Also around this time, it's probably likely that the abdomen would have started shrinking because, again, we know in all barnacles the abdomen is completely gone, and it's very likely that in this point we start to see a lot more hermaphroditism because organisms are really now settling into the sedentary lifestyle. From there, we move on to the extension of the peduncle and the beginning of the formation of shell plates. So importantly, the peduncle is just an extension of the head, and we know this because it still maintains the paired antennae at the very base of the peduncle. So you're just growing your head slowly, and this allows you to lift up out of the substrate. And if you've already begun to form these shell plates, then leaving the comfort and safety of your substrate is not harmful because you begin to have some shell plates. Importantly, though, we don't expect these early shell plates were very complicated. They were probably just a couple of them. They were probably abnormally shaped. They wouldn't have covered most of the organism. They would have just provided some initial protection to allow this escape from the substrate, right? And again, most of these organisms should have probably been well settled in to hermaphroditism by this time and should have all completely lost the abdomen. From this, we then move on into the modern Bilanomorphs. So the characteristic Thoracicans are the balanus, and they typify an extension of the shell number, fusion of those shells, and the settling back down onto the substrate by the reduction of the peduncle. Now, again, a lot of the modern ones still maintain the peduncle, so this would have been more of a split in which many kept the peduncle but increased shell number, whereas others began to reduce the peduncle as they increased and fused the number of shells and generated the shell wall. Almost all of these would have certainly been hermaphroditic, as we're now getting into the very modern types of barnacles. Now, again, this is a very cartoonish model, right? I'm just proposing possible ways in which this could have happened. Now what we need to do is look into the natural history of these organisms and see if we can find examples of each and every one of these stages. Can we support each one of these little proposed steps? Does it make sense? Is it plausible? Is it reasonable? So let's start off with this hypothetical free-swimming bivalved carapace ancestor. Do we have something that can help us think of what this might have been like? So remember when I said before that I wasn't going to tell you where the name cyprid comes from? I'm going to tell you now. So Cyprus is a genus of ostracods. Ostracods are bivalved crustaceans. That is, they have a carapace that is paired. They live within it. They have two antennae and very big thoracopods that they kind of kick to swim. As you can tell, this is exactly what the larval form of barnacles do. The cyprid. It gets its name because of its remarkable similarity to modern extant free-swimming ostracods. And they both also have gigantic compound eyes. Now, this is in no way saying that cyprids evolved from ostracods. Don't take that at all. What this is just trying to give you is an example of what that early ancestor could have been like, given that we have a larval form that very well represents what it could have been like, and we have modern organisms that very much represent the sort of niche that it would have been occupying, right? So again, this is just to give some support and get you to thinking about what that ancestor could have potentially been like. Second, now we're getting into a little bit more precise prediction. So the antennal attachment phase starts. So now we were beginning to get some sedentary lifestyle, but we need to maintain that carapace, maintain the abdomen, and be dioecious. Do we have anything that represents that? Amazingly, yes. And it just so happens that they are sister to the cirriped. These are the Ascotheracins. Now, the Ascotheracins maintain their carapace. Sometimes they even fuse it, and they attach just by their antenna. They don't form a peduncle, they don't form shell plates, nothing like that. They still have an abdomen, but they live sedentary lives attached by their antenna. In fact, they actually have a kind of hook on their antenna that helps them in their attachment. Now, most of these are actually ectoparasites. They live on sponges, echinoderms, and some corals. They'll just kind of like burrow and just like attach their antenna and then just live free floating on them. Some of them, however, have secondarily evolved endoparasitism, where they'll actually burrow a little bit further and live inside of their host. So again, this is just an example of what that ancestral condition might have been like that has been retained in the sister taxa of the cirripeds. And looking at this particular example of this Ascotheracid here, it looks a lot like the ostracods that we just looked at, right? It's not that differentiated from the cyprid form that we see in all modern barnacles. Okay, now let's move on to the next thing. So now we need to lose that carapace. We need to reduce the abdomen. We need to start burrowing because we've lost that carapace, and then maybe start seeing some early transition to hermaphroditism. Well, let's move one step closer. Now let's get into the cirripeds, but let's exclude the Rhizocephalans and the Thoracicans and just look at a group called the Acrothoracids. Now, the Acrothoracids shed their carapace, just like all modern cirripeds, but they don't build shell plates yet. All right. Instead, what they do is they burrow into rocks. They use those antennae to dig down into a rock, shells, corals, any kind of hard substrate. And then they just feed outside of that, the safety of the substrate that they've burrowed into. Interestingly, some of them have actually kind of strengthened their exoskeleton with these proteinaceous bars. You can see them on this example here, these little lines. They've just kind of strengthened it with a proteinaceous bar. So it's not super strong, it's not calcified, right? But it does give it a little bit of strength, given that it is completely shed off its carapace. Now, they're typically dioecious. But some of them do have they do show androdioeci, whereas the males are little dwarfs that will attach to and live on or inside the female as kind of like parasites, right? So we have now seen that we have examples of carapace maintaining organisms that are closely related to cirripeds that attach by antenna. We've now seen those that shed their carapace, burrow, but don't yet form shell plates. Okay, now can we think about what happened to that abdomen, right? As the prediction goes, we should start seeing the reduction of the abdomen. So a very fascinating study looked into the development of barnacles and specifically looked at the Hox genes, the developmental gene pathways in barnacles. And they compared these to the developmental genes in many other arthropods where we know where the abdomen forms. What they found, amazingly, is that two genes that are involved in abdomen formation in all other arthropods are broken in barnacles. In fact, they have repeat elements inside of those genes that have shut them off. Now, furthermore, they've even found vestigial abdomens within the naupliar form of barnacles, pictured here. So here's the thorax, and then here, this tiny little thing that's only present in the first couple of stages of naupliar development before it's just completely shed off during metamorphosis is the abdomen, this tiny little thing here. Right. So here we have very clear example of what happened to that abdomen, right? Again, if barnacles are crustaceans, they should have an abdomen. They don't have an abdomen, then we should find evidence that the genes, that the developmental pathway for the abdomen should be there, but has been lost. And that's exactly what we find. Those genes have been broken in barnacles. The abdomen is just a vestige in the naupliar form that is then shed. This is a comparison across the different groups showing this subsequent reduction in the abdomen. So looking at the Facetotecta and the Ascothoracids that both have robust abdomen, those are pictured in green here. ABD stands for the abdomen. You can see it here and here. We start to see the reduction of that abdomen, and the Acrothoracids, those are the ones we were just looking at before we got into the development here. So they still maintain it, but it's much smaller. And then by the time we get to the cirripeds, Rhizocephalans, and most of the Thoracicans, the abdomen is completely lost. So there's another way in which our evolutionary model has been able to accurately predict this subsequent slow and gradual decay, loss of the abdomen itself. Okay, fourth, now we need to start seeing the evolution of the peduncle as an extension of the head, as well as some early shell plates, right? We should not see the barnacle start to leave the comfort and safety of its substrate without beginning to get some of those shell plate formations. But importantly, they should be pretty simple. We don't expect to see really, really complex shell plates forming very early on. So to get into this, we need to dig a little bit into the dirt, right? We need to look at some fossil forms. What do the fossils tell us about the earliest barnacles that we see? The earliest barnacles belong to the group Phosphotathoracid. That's a mouthful, but it comes from phosphate and thoracids. And this is because they build their shells using phosphate instead of calcium, like all the other modern barnacles do. They only have 4 valves, these most ancient forms, and that leaves most of their body exposed. And importantly, these 4 valves are weird and like differently shaped, and they don't close completely. They're just a couple of little valves that sit around the barnacle. And like I've already said before, the peduncle is an extension of the head. We can see the antennas that are still expelling out of the bottom of the peduncle. And we also know the cement glands, which are what excrete the adhesive for barnacles to bind, are inside of that peduncle, right? Indicating that the peduncle is indeed an extension of the head. Now, I want to show you one of the very few surviving members from this phosphate group. And this is from the genus Ilba. It looks almost like a little chicken claw. It's this weird little critter, but this is the peduncle here. And then those are the shells, right? You can see they're oddly shaped. They don't even cover the full thing. You can see there's lots of open space between them. They live mostly in the deep sea, but they are the only surviving member of this group. And interestingly, they build their shells from phosphate instead of calcite, as I just said. Why? Why is it that they build their shell from phosphate? I'm going to give you a little bit of a bonus that was not part of our prediction, but was something cool that I came upon while I was researching this. So, during the Paleozoic, when we first began finding barnacles, and when all of the Phosphatothoracids first began to appear, the amount of phosphate was much, much higher than it is today. And in fact, this is the phosphate building shells of barnacles are not just restricted to barnacles. Lots of other organisms that built shells also used phosphate during this time because it was so abundant. But by the Mesozoic, the amount of phosphate in water just plummeted. And so what ended up taking its place was calcite. And so you see in organism after organism, all of these different taxa transition from using phosphate to building their shells with calcite. Right. So now all of the modern barnacles, with the exclusion of like just a couple of really, really rare deep sea forms, build their shell with calcium instead of phosphate. And I thought that was a really, really cool thing. As I was studying this, I was like, why are all of these ancient forms that arose in the Paleozoic, why are they using phosphate instead of calcite? Like, what's the reason for this? And I started thinking, well, maybe phosphate was more abundant. And I found this paper that sure enough, phosphate was much more abundant in that time than it is today. So that was a really cool thing. Just a bit of a bonus, was not necessarily part of our evolutionary model. So continuing on, as we see the more and more modern forms, we see more and more shells beginning to form, these shell plates beginning to form. They're still kind of random. They're not really quite fused like they are in the Bilanomorphs. They just are like spattered around the organism. Many of the gooseneck barnacles still maintain this form, whereas a couple of them have actually started to fuse those shells. And then if we look at some fossil forms, we can see this progression relatively clearly. So starting down here, we're in the lower Jurassic, and then working up to the Neogene, you can see that most of the diversity of barnacles has started to arrive by the Paleogene and the upper Cretaceous. So starting down here, these are the phosphate-only forms. These are the ones like I showed you before that have relatively simple shells, no more than 4 shells. And then as we start moving up, we start getting lots of various configurations where they're adding tons and tons and tons more shells. There's this interesting trend in the fossil record of first adding a lot of little shells and then progressively fusing those shells into bigger and bigger ones that better enclose in the entirety the organism. So by the time you get into the late Cretaceous, you start actually finding the Bilanomorphs, the most highly derived forms. Importantly, they do not appear anywhere in the fossil record prior to the Cretaceous. And this is exactly what we would predict. They are the most derived, the most different than any of their relatives, and so they must have arisen much more recently as a slow accumulation of the fusion of these shell plates and then the eventual loss of the peduncle. So that leads us right into the last part of our little evolutionary model: loss of the peduncle, fusion of shell plates, and the emergence of the modern Bilanomorph. So as I was just saying, Bilanomorphs are the youngest cirriped order, only appearing in the Cretaceous. They completely lack this peduncle. They just don't grow it anymore. It just stays flat. And the reason for this, right, is that they now actually form a fused outer shell. So you don't need to be sitting on a long stalk if you can just build a wall around yourself, right? And that's literally what they do. They have this fused outer wall that they sit within. And this has enabled them to occupy some of the harshest niches in marine systems, and that is intertidal zones, specifically rocky intertidal zones. We have really strong wave action pounding against the rocks, against the shore. Barnacles do exceptionally well, and they do so well because they're protected within these really, really tough shells. And then finally, they've specialized their interior 2 or 3 cirripeds as additional mouthparts. So this is unique to the Bilanomorphs. The other ones don't do this. They still have their back legs that are used for like wafting in the water to filter feed, but the front couple of thoracopods are reduced, and they actually are used as mouthparts to help sift through things. So this is an additional derived characteristic that's in the Bilanomorphs that's not in the Scalpellomorphs. Okay, so out of all of that, there is one little loose end that I feel like the model didn't accurately capture. And I was sitting around thinking about this, and so I wanted to present it to you. How does copulation evolve in a sedentary species? So think about this. A species becomes sedentary, and to reach one another, barnacles famously have really long penises, right? So they just reach up and they can inseminate their neighbor. But they didn't have very long penises ancestrally, right? That's a derived characteristic of a sedentary lifestyle. But we're proposing in our model that sedentary lifestyles evolved first and that a long penis must have evolved after this, right? So this creates a kind of chicken and an egg. How did that then arise? How were they able to fertilize their neighbors before they had the long penis to reach them? Kent kind of poses this question in so many disconnected ways, but that's effectively what he asks. And I wondered the same thing until I found this really interesting paper. So one of the most common barnacles are capable of copulation and broadcast spawning into the water column. So they will literally just spray their sperm into the water column. It will then settle on their neighbors and fertilize them. So they didn't need to have the long penis to be able to reach them if they could just broadcast their sperm. Lots of organisms do this, from sponges to corals to polychaete worms. Tons of organisms broadcast spawn. That is not a strange thing in the water, right? But had not really been recognized in barnacles because we thought we understood that they have these long penises. It must be because they only do an internal fertilization. But it turns out they do both. They will also just spray their sperm into the water column, which is probably the ancestral way in which they fertilize their neighbors. But you can imagine an evolutionary pressure that this places. You can do a lot better if you can just extend your penis to reach your neighbor. right? You more ensure fertilization in that way. So the pressure is on to evolve that, but in the meantime, you can still reproduce because you can broadcast spawn. So that particular loose end is nicely tied up into a bow by simply broadcast spawning. All right, so how did our evolutionary model do? Is it plausible given the natural history of crustaceans? I think so. I think given that each one of their more and more distant relatives show each one of these stages, I think lends credence to the model, right? We're not proposing, none of these life stages are outside of the realms of anything we ever see in crustaceans, right? Each one of these life stages are represented by extant organisms that do just fine, right? That live their lives perfectly fine, and so didn't need any magical steps between them that they would have had to overcome some wild thing, right? Extant organisms show these life stages. I think it's supported by comparative anatomy, in the sense that when we look at the internal anatomy of a barnacle, we see that they really are upside down, head first shrimp, right? The internal anatomy is very well supported there. And the internal anatomy of each one of their relatives is supported, right? We can very see the close homologies between them, especially when it comes to their developmental biology, right? They all develop in virtually the same way. It's the last adult form that is the most modified. Is it supported by the fossil record? I think so. The fossil record is a little bit sparse with some of the Acrothoracids and the Ascotheracins. These are the ones that are showing stages 2 and 3. Stage 3 is supported by marks in the shells and in rocks. You can find them by just looking at these little burrows. But lots of things cause those little burrows. So, it's up to interpretation whether or not that was caused by, an Ascotheracid or something else. So there's some debate there. But in terms of the formation of the peduncle, very early shell plates, not looking anything like modern shell plates, becoming increasingly complex, and then fusing, that's all supported by the fossil record. We do see that emerging pattern. Molecular biology overwhelmingly supports this, right? When we look for Hox genes, we can see very clearly that there are homologous genes in other arthropods for the development of things like the abdomen. It's just been broken in barnacles, right? They don't form it because it's broken, and we can see in their relatives a very clear successive loss of the abdomen, where you have fully formed abdomen in the Ascothoracids. The Acrothoracids have slightly smaller ones, and then the Thoracids have no abdomen, right? This is exactly the kind of successive loss that we would expect to see. With all of that said, let's take a step back and ask, I think, an important question. Is it true, though? Now, remember when I said in the beginning that no evolutionary model is true in that it captures exactly what happened? It doesn't even try to. And this is important, and I want to dig into this in just a little bit more detail and talk a bit about some of the philosophy of science. So to set up this discussion, let's think about a kind of simple hypothetical. And the reason I bring this up is because very often creationists will say things like, oh, but that's just your interpretation of the data. You didn't actually see this, right? It's all about your worldview and the way you're interpreting facts and blah, blah, blah. So let's start off with a simple demonstration as to why this style of logic is faulty when applied to science. So let's imagine I have a ball and I drop that ball. And what we observe is that the ball falls, right? And I ask you, why did that ball fall? You would say gravity. But notice that's an interpretation, because the only thing we actually observed is that the ball falls. For thousands of years, things have fallen, and nobody said, well, that was gravity, clearly. We observed it. There it is, gravity. No. Gravity, importantly, is the explanation. It's the model that is explaining why the ball falls. right? It has both a theoretical underpinning, that is to say that it's a gravitron that is interacting with a Higgs boson field. Like, that's the theoretical underpinning. It has a mathematical underpinning where we can mathematically describe why objects of smaller mass are attracted to objects of bigger mass, right? But all of those things are interpretations. We are explaining a simple fact, which is that a ball will fall when you drop it. That is the only data point we have. We are explaining that data point with a model that makes testable predictions. Science is all about the observation, which is completely devoid of explanation, then applying an explanation that can be tested, right? So gravity is an explanation for a very simple observation of a ball falling when you drop it, right? It's an explanation that makes testable predictions and that is falsifiable. Evolution is exactly this same thing. This whole evolutionary model I just gave is exactly this same thing. You could very easily falsify this model by just never finding at all there being an abdomen gene, or finding that actually when we sequence the genome, barnacles are not closely related to copepods or malacostracans. They're actually far more distantly related to that, and then therefore don't cluster within crustaceans at all, right? Like, we could set up experiments to test this hypothesis. I just want to make it very clear to creationists that if you accept gravity as an explanation, as a model to explain a ball dropping, and you don't call that a religion, then evolution follows the same logic, right? It's the exact same logic. Evolutionary models seek to explain the things that we've observed, that inheritance always works, that populations always change, that DNA is the material of inheritance, and when you take all of that together, you create a tree of life that sure as heck looks like everything is related, right? And that is the interpretation of the data. If you have a better interpretation, put it forward. Explain when DNA stops working. Explain when populations stop changing. Show us where that line exists on the tree of life, and get it published in a peer-reviewed paper, and we can talk. I think a lot of it boils down is that creationists don't even really seem to have a firm understanding of science, or they have this warped view where they change the definition to fit their own needs, where we're talking about a worldview when we're talking about evolution, but it's complete science when we're talking about gravity, right? Despite the fact that when I say all organisms share a common ancestor, they'll say, well, you're using a worldview interpretation. But if I say gravity explains why a ball falls, you'll say, yeah, cool. I'm with that science. Let's talk a little bit about what science actually is, and try to like enunciate a little bit on the limitations of it, and why really and truly we shouldn't try to pit things like evolution and creationism, right? Because creationism doesn't exist because of science, right? That's not why it exists. It doesn't exist because of evidence. It doesn't exist because it makes accurate predictions about reality or what we should expect to find in the dirt or in your genome or anything like that. It exists because it's a religious belief. It exists because there is a thing called the Bible, and there are people called Christians that accept that the Bible exists, right? Like, it emerges from a religious belief. Science is completely different than that, right? These two things are diametrically opposed to one another, right? And so by pitting this evolution versus creationism, we're not arguing on this about the same thing. We're coming from different foundations. Science relies on methodological naturalism. Creationism doesn't. It doesn't rely on naturalism at all. In fact, it rejects naturalism like in its totality, right? So you can't make a scientific claim about reality while fundamentally rejecting naturalism, because then you violated science. You violated the fundamental assumptions of science. So let's talk a little bit about what those assumptions are. Firstly, science is a philosophical system of knowledge acquisition and organization about the physical world. right? It proposes testable explanations about current observations that are predictive of future observations. All knowledge gained by science is tentative on future observations. Everything is tentative. This is why I said any evolutionary model that I could propose about barnacles is not true in any absolute sense, with like a capital T true sense. It's only true, it's most supported by the current data that we have, right? Now, if you have a different interpretation of that data, right, and you think you can propose a model that better explains that data, but that is also falsifiable and that also follows methodological naturalism, then you have a seat at the table, right? But if your model cannot be falsified, if there is no data out there that will overturn your model, right, that you can't just reinterpret to fit your model, then you're not doing science. Right? You're fundamentally not doing science. And that's not an attack. That's not, I'm not saying like you're not doing science or get out of here. You're doing something else. You're doing theology, and that's fine. Theology has its own place, right? But it's not in science. Science doesn't belong in theology. Theology doesn't belong in science. These are non-overlapping magisteria, right? But I want to end this on just pointing out that many scientists and many people that argue, science are forgetting that there are 4 fundamental assumptions of science. That if we're going to do science, that if you and I, whether you're creationist, evolutionist, like whatever your background is, if you are going to make a scientific argument, there are 4 assumptions you must make. You have to accept these assumptions. Now, that's not to say you have to accept them in all walks of life, right? It's just to say when you're doing science, to do science, the scientific method relies on these 4 things being true, all right? And they are unknowable and they are untestable. There's no way to know whether these things are true. But these are the 4 things, and that's all that science relies on, is that these 4 assumptions are true. 1, that there exists an objective reality that we all experience, right? That the physical world that I experience is the exact same physical world that you experience, okay? Second is that this objective reality is governed by natural forces. When something happens anywhere in the universe, it happened because of natural forces, right? Now, again, this is not to say that supernatural forces didn't do it, right? That's not what that means. It just means that if supernatural forces did do it, science will never know. It will never be able to get the right answer because it can't investigate that question, right? It can't use the supernatural as an explanation for any observation. It can't, right? That violates this assumption of science. Third is that the natural forces are discoverable by humans. That is to say that our intellect is actually capable of understanding them, because there could be an objective reality governed by a natural force that is so beyond our understanding, we'll never know it. We just can't intellectually conceive of it, right? If that's the case, we can't investigate it. It's beyond human knowledge and therefore is beyond the realm of science. Lastly, and this is the one that creationists overwhelmingly reject, but is very, very important, is that the natural forces that govern this objective reality are uniform across space and time. So whenever they occur, wherever they occur in exactly the same way, right? And this is important. If I'm going to make a prediction, if I'm going to look at an observation, propose some natural force that explains it, and predict future data, that natural force can't just change its mind tomorrow and do something else, right? That natural force couldn't 1,000 years ago suddenly decide that, no, I'm going to decay at a faster rate, right? Like natural forces are uniform. They have to behave in exactly the same way. When they stop behaving in the same way, they're no longer predictable, right? And if we can't predict them, we can't know them. And if we can't know them, they're not part of science, right? So for us to investigate a natural force, it needs to be uniform everywhere that it has ever occurred and for all time. This is why I really hearken on the point that we understand inheritance and we understand that populations change, right? And that assumption is true so long as there's life on the planet. If there's life on the planet, those two things are true, right? To say that they aren't true at some arbitrary point in the past, as I said, is to violate this fourth assumption, right? Is to violate the assumption of the uniformity of natural processes and natural law. So you would then need to explain some new natural force that replaces that one at this point, at this time slice. At this point, we replace this natural law with some other one, right? And again, this is very important that we understand these are the philosophical underpinnings of science. You can reject any four of these in your day-to-day life. You can say, you know what, there's not really an objective reality. There's actually a bunch of things that you and I experience differently and we don't see the same of, et cetera, et cetera. But when you do science, we have to assume that an objective reality exists, right? It's a tentative assumption while you're doing science. When you walk out of the lab, you can reject all four of these things. It doesn't matter, right? But to do science is to assume methodological naturalism and to assume these four principles. So again, I give you all of this as again, sort of a way to tie all of this together, because I already know that no creationist is going to watch this and be moved by it. None of them. They're going to see my little cartoon. They're going to listen to my, if they even get that far into the video, because it's like 40 minutes in. If they even do that, they're not going to be convinced by it. They're going to say, you didn't observe that. That's what they're going to say. You did not observe that. That's a fairy tale. You made that up. Without understanding how testable explanations work, without understanding how hypotheses work, without understanding that you don't observe gravity when you drop a freaking ball. You're observing a ball falling. You're accepting the explanation that that ball falls because of gravity, right? So you can't apply that to one field of science, biology, but like not apply it to another field, physics, right? It's important you be consistent. And so I already know what they're going to say. None of them are going to be convinced, but I really hope that maybe by talking a little bit about the philosophy of science, by giving a little bit of a primer on evolution, that they maybe learned a little something about the philosophical discipline of science, about where evolution falls into that, how it fits in with all the other realms of science, and honestly, that you learned a little bit about barnacles, because like at the end of the day, we're really here for the barnacles, right? They're the coolest thing. So I hope you enjoy the video. Please leave me a comment, give me a like if you like it, and I'll see you next time. Thanks.